are back with 3 other startups ready to present their 5-minute pitches. And after the 5 minutes, there's time for Q&A with the jury. So far, we've seen very high quality pitches and it's been very entertaining to watch. So well done. I hope that the presenters for the next round are ready to go. First up is Lenio Bio. And Lenio Bio is disrupting the way proteins are made. Their cell-free protein production platform is a pandemic preparedness platform and useful in a broad range of other applications as well. Joining us today is CFO Francesca Finke and Ricarda Finan. Francesca, go ahead.
Thank you very much. You see the presentation already, or?
Yes, I think we will move on to the next slide. There we go.
Okay, so welcome everyone. My name is Francesca Finke. I'm the CFO of Lenio Bio, and I'm very excited to share an impression of Lenio Bio today and to represent our leadership team. So we have accomplished great success in the fight against deadly diseases. Tetanus, polio, or pox virus have been erased completely in many regions of the world. But then from time to time, we get surprised. Pandemics can hit us out of the blue. Most prominent right now, of course, COVID-19, the reason for us to hold this meeting today virtually. So if we have such powerful tools and technologies, why does it still take so long to react on a pandemic like COVID-19? Promising vaccines are based on proteins, and proteins are mostly produced in whole cells. But cell-based systems are inflexible, time-consuming, and expensive. Candidates fail during the development process, and only a fraction can really be taken to the market. It is the cell line optimization that costs most of time and money, because several cell lines are needed for different types of proteins. 20% of candidates cannot be extracted in cells at all, and multiple steps are needed to optimize the process. To fasten the process, we need better technology. At Lenio Bio, we are challenging the industry standard and want to change how proteins are made, make it faster, simpler, and more cost-effective. Our selection for the Horizon 2020 SME instrument last year enables us to follow this mission. Our ELIS technology is disrupting the current industry standard. ELIS is cell-free and simple, using only what is needed. From the plant cell, we remove debris and enzymes, and we really only keep the machinery of the cell that produces proteins. You then just add the DNA of the protein of choice, and after 24 hours only, you have your protein. ELIS is an open system, so of course conditions must be adapted depending on the protein, but it can be done easily outside the cell. This optimizes expression yields in no time, unlike to every cell-based system. The expression yield for insulin, for instance, is in ELIS 6 times higher than the current industry standard. ELIS is further able to produce hard-to-express proteins, so different types of proteins are ready in just one platform. So in total, ELIS can really shorten the time to market for protein-based drugs and vaccines massively. The technology is patented by Corteva AgriScience, and we hold an exclusive license to market and develop the platform. In 2018, we launched the ELIS kit, which can be used as a screening tool for R&D. We complement the kit with our protein services. By end of 2020, we will launch ELIS as bulk material, so enough for customers to go into clinical trials. And by end of 2021, our pilot production line for kilogram production of proteins will be finalized. So ELIS can then really be used throughout the whole development cycle of protein-based drugs. The ELIS kit and protein services generate revenue already, but more important, they also open doors for future licensing and partnership agreements. So ELIS can really be a game changer, not only for big pharma, but for a variety of other applications, like sustainable food, personalized drugs, or synthetic biology. There is an immense potential. And beside the food business, where we receive a lot of interest from the market right now, we aim to introduce ELIS as a PPP, a pandemic preparedness platform. But disrupting a current system is very challenging. So we need to strengthen our sales activities to gain visibility and sustainable revenues, create the necessary robustness of the system, and get GMP ready. And we therefore need an investment of 6 to 10 million from long-term investors who can help us address these challenges. So imagine that we can change today's standard technology and shorten time and cost to market for protein-based drugs massively. With Alice, we can do that. So let's go and save lives now. Thank you very much.
Thank you very much. Well done and well within time as well. Let's switch over to our jury to see if they have any questions. Who would like to take the first one?
I have a question. Thank you, Franziska. So for the proteins to be used as therapeutics, you mentioned that certain partnerships may be established to test whether proteins produced with the Alice platform are effective as therapies. What strategies are being taken there? Are you going for off-patent existing therapies, so doing biosimilars? Or are these new therapies, like new antibodies or other proteins that are currently under development?
Yeah, maybe Ricarda, so I have our CSO, Ricarda Finnan, with me for the more technical parts of the questions. So maybe you can say something to that as well.
Yeah, definitely. I think like with our technology, we can really benefit partners along the whole value chain. And then also we are looking into the future. So with new antibodies, it could be biosimilars, but also other formats, really, we can already collaborate with partners from the discovery through the early development, but then also the later development. But of course, there are also options of like antibodies also that did not make it through the clinic that can be perhaps had production problems, can be produced in the Alice system.
Do you foresee that there would be any challenges in demonstrating bioequivalence if you're using a completely different cell-free platform?
We haven't done it, so we don't know it. We don't think so, because it's easy to really characterize the compounds that are coming out of our system. And the question is then, is it like a bio-better even biosimilar? But this is something we would have to test really on a case-to-case basis. But definitely we could show already that antibodies can be very well produced within Alice.
I have a question from Laura as well, and Cem and Eric are waiting as well, so please keep it brief.
Yes.
Okay.
Laura.
Very quick technical questions, please. Do you have a limit in the size of proteins that you can produce? And are there differences in modification, protein modifications that are that occur through your plant system compared to other cell-based?
So the largest we have produced so far are like antibodies, 150 kilodaltons, some enzymes that have like in this size. So we did not push it to the limit. And we do have post-translational modifications. So really have glycosylation and disulfide bonds for functionality as well.
Thank you.
Okay, let's move on to Eric then.
Yeah, hi. You said you have an exclusive license from Dow. Can you tell us about the terms of that license?
What terms of these licenses?
Well,
so
royalties,
yes,
milestones, yeah.
Yes, we pay royalties, and Dow really protects the patent. So whenever someone is about to infringe that patent, we really have a strong partner there, and we pay royalties based on one milestone, and then based on the revenues.
What percentage?
Sorry?
What percentage you pay of your sales?
The percentage.
One digit percentage, yeah.
Okay.
Then we move on to Cem.
Thank you. Just a quick question. I mean, what are the risks that you see going forward, and what's the plan for managing those risks?
I think from the technical perspective, we are quite on track, because the risk is really that we have to scale the technology, but there we really reached some of the most challenging milestones, I guess. Then a risk I see is that we really need to strengthen our sales activities and become or get to the point that we can carry the activities on our own. So we really need to strengthen our business development and our sales activities and marketing. Yeah, and then, as I said, also there is a huge variety of applications that we can potentially target, but we really need to identify the most valuable markets for us. So right now, we did that in the vaccine business, as I said, to introduce Alice as the PPP, and also the food industry, because we can produce quite well growth factors that are needed in the artificial meat business. So these are the first things that we are still evaluating, but I think there's a risk of distraction that that's challenging us right now.
One final very quick question, maybe from the jury. Or otherwise, I guess we're almost at 5 minutes as well. Killian, did you have something to add? Was that it? Okay, I think that was it for now. Francesca and Ricarda, thank you so much for your pitch. Lenio Bio, as they mentioned, they're looking, among other things, for a strong and long-term investor. And it's good to see some answers to the question that I think all of us have right now. Why does it take so long to make vaccines? Well, Lenio Bio has a solution to that, and they're looking to go to market. And in 2022, I'm saying that correctly, it should be available for the whole cycle. Now, the jury is scoring this pitch right now, and we're getting ready to go to our second pitch of this round. And we'll be joined by Medetia. And Medetia was founded in 2019, and the team is developing first-class treatment for ciliopathies. It's a range of rare diseases with a high unmet need, and they are developing and discovering drugs through strong partnership with the Imagine Institute. Now, please welcome Jean-Philippe Anero. Good luck on your pitch. Ready to go. Are you there? And is your camera on and your sound on? Because then we can move on to Jean-Philippe from Medetia. I hope he's there. I'm going to check with our technical crew to see if anything is going on. He's having some issues, I see. Now, meantime, while Jean-Philippe is getting ready to get his pitch going and present to us and use his 5 minutes, I would like to mention to you again that we have our matchmaking platform ready to go. If you have some time during one of the breaks, you can take a look at the platform, schedule meetings, and also make sure to check and see if you have any pending requests from others. You can meet the delegates there, the startups, of course, and other investors who might be interested in your company, or just to meet up and see what could happen in the future. There will be more time during lunch, in which we will be having about 30 minutes after this round. Then there will be another pitch round, and at 5 p.m. today, we will go towards our awards ceremony. And then the finalists for December will be announced. And also we have some wonderful sponsor awards to award to some of the competition today. Now, let's see if Jean-Philippe is ready to join right now. Let's try again. He's still having some issues because I don't see him joining yet. Let's just go over the startups that we have seen so far. In the first round of pitches, we've seen Double Bond Pharmaceuticals, and they've been developing a novel gel formulation that can help prolong patients' life who are suffering from brain tumors. Very important development as well. After Double Bond, we saw Gelmatic, Dr. Lindemann, who was explaining to us that they have developed also a gel, but for a very different treatment, namely lower back pain and osteoarthritis. There's a disc disease that many people suffer from, and this gel could make a very big difference in their lives. Good Gut, a pitch that we saw in the first round, they're focusing on colorectal cancer. Nowadays, it's an issue that many of the patients who are being diagnosed with this type of cancer do not get the right type of testing, or the tests do not get the right results. So they've made a solution for that is non-invasive and also a lot cheaper than the current type of tests. Then we saw HiQ Nano. Mauro Molinetti was presenting from them. And they're making out-of-the-box approaches in the field of home care, and they have a test kit that you can try and find out what kind of antioxidants you have in your body at home instead of in a lab. The test was called iBlue. I think we may be ready to have Jean back right now. So we're going to switch back to the pitches. Let's see if Jean-Philippe is there from Medetia. Can you hear me?
Yes, I'm here. Could you hear me?
There you are. Yes, you're all good. Ready
Okay,
to go?
thank you.
Because then we'll start the clock for your 5 minutes.
Okay, Thank you very much. My name is Jean-Philippe Anoro. I'm the CEO and the co-founder of Medetia. So Luis Briseño and myself are very happy to present you our strong will, our ambition to treat patients with severe nephropathy. Next slide. Next slide, please. So as you see here, this is Jeremy, an 8-year-old boy, and his boy is suffering from renal nephropathy. For this Jeremy, there is no other option than graft transplantation. Jeremy is suffering from, next slide, nephronoptosis, which is a genetic disease, a severe pediatric nephropathy that is caused by the gene deletion. And this deletion is inducing the absence of the cilia, and this cilia absence is causing the damages of the cilia. Our will, our ambition is to provide therapeutics that will restore the cilia and then restore the tissue function of the kidney. Next slide. So this is a huge medical challenge, and this is the reason why Medetia decided to locate its headquarters at the Imaging Institute, a world-renowned institute for genetic diseases. We have now an expertise team on cilia, but also an expertise on the clinics, with the director of the Imaging Institute at Hospital Necker and also the researcher that discovered the gene. Next slide. Our proposition is very clear. We have now some compound, MDT-109, MDT-110, which is compound that will provide therapeutic advances on nephronoptosis. Those compounds are already used in phase 2 for other diseases. Those compounds have been validated on NPHP1 murine model that recapitulate the disease, and also those compounds hit pharma target already known and validated for other diseases. We have also secured our path and secure some backup approach with a unique discovery approach using patient cells and microscopy. Next slide. Next slide, okay. So our objective is to propose the first-in-class orphan drug to serve this high medical need. The population is identified. This is the pediatric patient bearing the mutation NPHP1, and also 1% of adults suffering on end-stage renal disease or late onset of nephronoptosis. The benefit is to provide an oral compound to avoid the burden of the dialysis and also the renal transplantation. Already several help supported our mission, and we gathered not only investment for business angel, but also subsidiaries from many different support that exist in France and Europe. Next slide. We are now ready for a go faster plan toward the phase 2. We secure 1.5 million euro to end and complete the preclinical validation of MDT-110. And now we will engage in 2022 a 6 million investment to enter the clinical phase by de-risking and at the toxicology and regulatory point of view, the compound MDT-110, and preparing a clinical batch and entering the phase 1 studies. Next slide, please. Our position IP is very strong because we have the license, an exclusive license of the patent that we would like to use for those compounds, and also the freedom to operate on this compound. And there is no right of third party now likely to prevent the exploitation of MDT-110. Next slide. We have also secured a backup program by signing a unique partnership with Ictos, a medtech company expert in artificial intelligence in France. Next slide. Here is the development and funding plan. This development and funding plan is targeting a phase 3 in 2025. The market is identified. It's 8,000 patients that we expect from Japan, US and Europe. And because of the clinical benefit we can envision for this severe pathology, we can envision a high price. This is consistent with the high value across the time of this plan. I know this timeline are ambitious, but because our location at the Institute Imaging and because our preferential access to the patient, we strongly believe that we can help patients like Jeremy to avoid graft transplantation. Thank you very much.
Jean-Philippe, well done. And thank you so much for your pitch. It's time to switch over to our jury. Who wants to have the first question?
I'd have a question for that. Would this then be a chronic treatment that you're proposing?
Yeah, that's an excellent question. Yes, we were proposing a chronic treatment. We would like to reinduce the differentiation of the cell in the kidney. And this chronic treatment presents the advantage of using dose that we can space the dose to avoid any acute toxicity, and this is exactly the strategy: chronic treatment.
Okay, another question from the jury, maybe.
Yeah,
yes. Oh, sorry. Go ahead, Eric.
Yeah, sorry. Yeah, what's the size of the market you want to address here?
Excellent question. The size of the market is the size of the incidence of the NPHP1 deletion. At the beginning of the study, we believed that it was a rare disease, and later on, last year, we made a very deep investigation with the England Genomics Consortium, and we discovered that this is the most widespread deletion, gene deletion in humankind, one of the most widespread. It's 1 over 140 heterozygote incidence. So we have now identified 8,000 patients, but there is more patients since we are just focused on homozygote deletion, and there is also patient with heterozygote composite deletion, but those patients will be defined later.
There was another question I heard as well.
Yes, please. Jean-Philippe, can you tell me a little bit more? Because I got confused. You mentioned 2 compounds at the beginning, but the plan that you presented and the cost that you presented refers to one of the 2 compounds. Is that correct? Yes. So is this, maybe I missed it again, is that an injectable, an oral compound? How is it administered?
So far, there is no, we made a test on a murine model by IP, but there is no obstacle to have an oral treatment. This compound is soluble, stable, nanomolar.
All right, more questions.
I have one more question.
Let's go to Morten first and then
okay, very quickly, a little bit more about the competition in the market.
Yes, a very good question. There is no, I don't say there is no competition, but the only competition we can see is genic therapy today. And because of this is a systemic disease, there is mostly damage on the kidney, but the genic therapy would like to be a general treatment. And I think genic therapy is not ready for that yet. And there is no identified competitor of that. We strongly believe that we have an advance because we are already proposing a compound in phase that has been used in phase 2. So it will be a redevelopment of a compound that is already known from regulatory agencies.
I think I heard Anta as well.
Yeah, so just can you quickly explain the mechanism of action of the drug?
Yeah, so it's quite confidential, but we understand the way we understand the drug is that, of course, this is a gene deletion. So the drug is not hitting the missing protein. So the way we discovered the drug by a phenotypic screening, but after that, we understood how the drug is working. And the drug is hitting a receptor, an agonist receptor of prostaglandin. And this receptor is, by the cascade of the regulation, is reinducing a well-known process of redifferentiation of the cilia. So that is a functional completion by hitting a rescue pathway that reinduces the formation of the cilia. And this has been verified.
So it has regenerative function.
Exactly, a regenerative function of the cilia. And in vitro, we monitor the regeneration of the cilia. That's correct.
Okay, we have one more quick question, or maybe an additional question by you, Anta.
Yeah, I'm just thinking about your, do you have an in vivo safety with chronic administration given the regenerative capacity?
Yes, we treated the animals for 6 months, and because to observe this benefit on the kidney, you have to observe the animal for a long period of time because of the natural evolution of the disease model. So far at the doses where we see the efficacy in terms of reduction of the dilatation of the cyst in the kidney, we don't observe side effect on the animals.
Okay, and we'll leave it at that for the jury Q&A. Jean-Philippe, thank you so much for your pitch. That was Medetia, and they are developing like drugs for very rare diseases. I have to say rare because that's what they first thought, but apparently their group is much bigger than they initially expected. They already have gathered investments and also subsidiaries, and they're ready to go to the next stage. Let's see how quickly they'll get there. The jury now has some time to score the pitch before we move on to the next one. And we already saw him briefly here in the image. We're going over to George Frodsham from Medi Sieve. And Medi Sieve developed a platform technology to revolutionize the treatment of bloodborne diseases like COVID-19, but also leukemia and sepsis, through the removal of specific harmful substances directly from the bloodstream. George, ready to go. Good luck.
Thank you very much. So hi there, everybody. My name is George Frodsham. I'm the founder and CEO at Medi Sieve. And I want to start by asking you the same question that we ask clinicians. What would you do if you were able to remove any harmful substance you wanted from the bloodstream of patients? What diseases could be treated? What lives could be saved? That's our mission here at Medi Sieve, to revolutionize the treatment of bloodborne diseases. And when we put that question to clinicians, they come up with a whole range of potential answers for areas in which this technology could have a really strong impact. And we call this platform technology magnetic blood filtration. And it is similar to dialysis in the sense that we circulate a patient's blood through what's known as an extracorporeal loop. Into that loop, we infuse the Medi Sieve beads. These are magnetic particles coated with specific antibodies which bind to the targets in the blood. The blood then flows through the Medi Sieve filter, which captures the beads and everything that they're bound to. So from this, you can see that anything that we could target with an antibody can theoretically be removed from the bloodstream of a patient in a cost-effective treatment of 2 to 4 hours, and products which integrate with standard existing hospital equipment. And as mentioned, there are a large number of potential applications for this technology and some huge potential markets. But today, I want to focus on our first product, which is for the removal of IL-6, a key inflammatory cytokine which plays a major role in sepsis, and it's been discovered more recently, COVID-19. Sepsis represents a massive problem worldwide. In the 5 main markets in the EU, there are 1.25 million patients. In the US, 1.7 million. Sepsis kills 50,000 patients in the UK every year. That's the equivalent of a football stadium. And it accounts for 5% of annual US healthcare spending. Sepsis is a major human problem and a major economic problem, but therefore also a major commercial opportunity. And we forecast getting revenues of nearly £750 million within 10 years, with gross margins of over 60%. And we're going to achieve that by selling our suite of 3 products. The Medi Sieve Magnet and Medi Sieve Filter work together in tandem and are the same for every disease that we go after, whereas the Medi Sieve beads are tailored to the specific target that we want to remove, with the first one being for IL-6. The filter and beads are single-use consumables and the largest drivers of margin and revenue. While our product includes an antibody, it's important to understand that they are all regulated as medical devices, and we have had that strategy validated by the MHRA. We are going to parallel track CE mark and FDA approvals in order to reach both the EU and US markets quickly. Our technology is unique and has a number of advantages over what's out there at the moment. And in particular, you can turn it off. What this means is that we do not look to necessarily eliminate all of the levels of IL-6, but rather provide doctors with the ability to control them, to prevent immune overreactions whilst maintaining the patient's ability to fight the underlying infection. And we've demonstrated this technology in-house as being fast active and effective, removing over 80% of circulating IL-6 from human plasma in less than 20 minutes. We own all of our intellectual property, having secured an assignment deal from UCL upon spinning out. We have 2 core patents, both of which have been granted in the US and are pending worldwide. Right now, we need £5.5 million in order to deliver the pivotal clinical trials, enabling us to get the regulatory approvals to go after our key markets from 2023. We have already secured a grant of 1.5 million towards that amount. So today I'm looking to complete a round of 3.5 million, which will take us to a Series B growth round in early 2023. We have already raised £6 million towards this, including grants for some very competitive agencies, and have also won a lot of awards, including the prestigious MIT Tech Review Innovators Under 35 award just this year. We've also received significant coverage in the national press and work with some of the countries and indeed continents leading lights in these, both from a regulatory perspective, clinical perspective, and commercial perspective. We have some core partners supporting us to achieve our goals, working with our diverse team, including 5 PhDs. Thank you very much for your time and attention. I've been George Frodsham telling you about Medi Sieve and magnetic blood filtration, a platform technology with large markets looking to complete an investment round of 3.5 million. Thank you very much.
Thank you very much, George. I would say over to our jury.
Like the first one, maybe.
I hear Cem first, and let's go to Anta after that. Cem.
Okay, just a quick question. What will be the treatment cost? I mean, could you tell us a little bit about pricing and costs?
Yeah, pricing is always the golden question. So at the moment, we've forecast our revenues based on a cost of £1,500 for a single treatment for a sepsis patient. And that's based on what's done in the market at the moment and our healthcare economics modeling, which shows that at that price, we can actually reduce the overall cost of treating these patients through resolving their symptoms faster and getting them out of the ICU.
All right, and I heard Anta as well on the phone.
Yeah, so I was just curious to understand a bit the magnetic blood filtration approach. Do you have any in vivo data that show how the selectivity of the approach?
Yeah, so the selectivity of our approach is linked to the selectivity of the antibodies that we're using on the surface of our beads. And so therefore is something we look at right at the beginning of product development to ensure that what we're removing is exclusively what we're actually trying to remove. So that's how we underline the selectivity and specificity. So we can be sure that we're only removing, in this case, IL-6, and for our future products, only, for example, CD19 positive cells.
Any more questions?
Can I expand a bit on that, or is there anybody else? Sorry.
Did you hear that correctly, or can you repeat it, Anta?
No, I heard it. I was just wondering the data you have in vivo. Can you expand a bit on the you have an in vivo preclinical proof of concept here, or?
For our bead data, we've only got in vitro data, but we have demonstrated specific removal from human plasma. And also we've got some early blood data as well, which shows that we're not removing any components that we don't want to remove. And of course, that's data that I'd be happy to share with you afterwards in more detail.
Thank you.
Yeah, it's Eric here.
I hear Laura and Christoph. Christoph, go ahead.
All right, thanks a lot. I have a question on your plan on reimbursing your product and maneuvering your way into guideline treatment. So what's your plan there? Because in the end, you're removing a cytokine, and you're not actually removing the cause of the disease. So maybe these are even 2 questions.
That's absolutely correct. I'll actually deal with the second part of that first. The removal of the cytokine is to treat a dysregulated immune response, which you're right, isn't really the disease, but it is the key condition. You know, that's what sepsis is, for example, and the driver ultimately of mortality in these patients. So the objective of bringing those levels down to normal is absolutely medically and clinically rational. In terms of reimbursement, that's actually something that we're looking at in a lot of detail at the moment, and there is some predicate work already done. So a competitor Cytosorb, who has a non-specific approach to this, has reimbursement in Germany and Switzerland surrounding the removal of cytokines through their non-specific absorption column. And that validates the sort of reimbursement route that we could take to enter into those markets. And that's what we're looking into right now.
I think I heard Laura as well.
Yes, please. Hi, George. I wanted to ask you if I understood it well, your IP covers basically your magnet, your filter, and the beads would be different in terms of depending on the application that you want to apply your system to, right? So is the patent covering all types of applications, or do you have divisional patents directed to the different applications with different beads?
So the most important patent we have is around the magnetic filter. And because it's the same device used in all applications, it's an incredibly broad patent, which means that it protects all disease areas. So it's very difficult for anyone to enter the market and do exactly what we do because of the breadth of that patent, which has been granted in the United States. In addition to that, you may have noticed I was very light on the details on exactly how these beads are made and what they're made of. And that's because at the moment we're looking at getting a similar generic patent around the way in which that operates. I'm going to be careful what I say there, but certainly at the moment we've got something that protects all of our markets for the magnetic filter, and we're looking to double down on that with an additional patent, which we're drafting at the moment.
Time for one final question.
Yeah, Eric here. You said that you're expecting to you have a huge target in terms of revenue, but at the same time, I mean, the methodology you propose is quite heavy because it's extracorporeal therapy. So is it consistent?
Absolutely. Extracorporeal therapy is very common in ICUs. I mean, obviously dialysis is the most famous, but ECMO, hemofiltration, and various others are already there. And these target patients for sepsis are in the ICU. So that equipment is available. These treatments are routine. And, you know, we've been doing extracorporeal therapies for over 60 years. So while it may seem strange, that's actually one of the less challenging areas of our technology.
George, so much for your answers. Thank you, jury. I'll leave it to you to start scoring, and good luck also to these 3 pitches that we've just heard.